Assange, everybody, and welcome to this session. I hope you're enjoying the heat. But today it gives me great pleasure to talk to you about some of the research that I'm quite interested in, and I'm trying to pursue in that area as well. And that is looking and trying to understand the designs in living systems. So just very quickly, a little bit about myself. Musavar just asked me to give a little introduction about my background, just so that it helps the audience to understand where I'm coming from, what I've had to learn over time, and how I've come into this area in the first place. So my background is actually in mathematics. I did my undergraduate at Imperial, but I gained a strong interest in mathematical biology. And that is the development of models to understand how living systems work. Then I did a discipline hopping PhD in systems biology. And that was quite a unique learning experience, because that was the first time I was ever exposed to working collaboratively with many different types of researchers, both on the modeling side, that is the theoretical side, and on the experimental front as well. And I was very fortunate to have the opportunity to collaborate with a group in Kyushu Institute of Technology in Japan. I visited them there as well and learned a lot of stuff about modeling and about the experimental side of things. But I continue to pursue my fascination with how bacterial metabolism works as a postdoctoral researcher. And I'll come on to defining what metabolism is in a moment, but I looked at different aspects of it and in collaboration with researchers from different departments even, ranging from, I hope you see my laser pointer here on the screen, ranging from microbial sciences to plant sciences, maths, and even engineering and chemistry. Looking into the evolution of metabolism, the regulation of it, and how it controls that, and how we can re-engineer that control. So my fascination for understanding this regulation of metabolism as re-engineering lies at the interface between many different disciplines, from chemistry, maths, and molecular biology. And the fields of research that I'm striving to contribute to include systems biology, metabolic engineering, and synthetic biology. That is how to understanding the biological systems and how to engineer them. So I think in this talk, I would like to show some examples of our understanding, or at least of my understanding, of regulation and control systems in nature, including from those examples of my own work, and how the systems seem to be designed beautifully for their roles. I want to discuss some of the visions of the researchers looking at how to re-engineer these bacteria for applications that will benefit us and our environment. But as the aim of the cafe, and as I go through the talk, I would just like to ask you all to bear in mind the strong interdisciplinary nature of the examples that I will touch on. And what I mean by that is that it's really the integration of knowledge from different disciplines, not just from different researchers, but researchers that have used this integration of knowledge to help us to give a glimpse of nature's workings. And at the end, I would like to pose some questions and get your own opinions, create a sort of discussion on the impacts and worries of engineering living cells. And I'll touch also on some verses of the Holy Quran that have sort of inspired me to take my view on how we should go about engineering living cells, or if we even should. So, since all my examples of designs in the living cell will center around the metabolism of bacteria and its control, I want to start by giving you a quick introduction and very high-level view of what's going on. What is this? What is the metabolism? So let's look at one of these E. coli cells that you see on the left-hand side, on the right-hand side here. So the bacteria are some of the simplest forms of life on planet Earth. They're composed of a complex network of chemical interactions, chemical reactions, and this are known as metabolism. These can break down nutrients into chemicals that form the DNA, that construct the cell's machinery. These are the proteins that can express the genes, that can catalyze, that are the enzymes to catalyze all of these chemical reactions that are happening in the cell, and to generate the energy needed to power these machines. The metabolism also constructs the building blocks for the cells to grow, and even produces chemical byproducts that can even create the microenvironment for the cell to live in. But it's very costly to produce all of these proteins, and not all of these enzymes should be made, and not all of metabolism should be on. And there needs to be an investment in some proteins which are regulators, which are controllers. And these control which reactions are on or off. So if you look here towards the bottom center, you will have these proteins which are deciding to turn on or off, some reaction, some part of metabolism, if it's needed or not. And these are the cell's control systems. But what is a control system? So for those who are wondering what that is in the first place, I'm sorry I'm going into such basic detail, but I thought this was a really nice analogy. And you think about your thermostat at home. Yeah, there's heat produced, and the thermostat senses through a sensor how much the temperature is, compares it to the temperature that you've set it to, and it decides whether to increase the heat or increase the heat or leave it off, depending on what that temperature is. So there's a set point, and the inducer sort of fluctuates over time until it converges to that temperature that you've set. So the analogy in the biology, if we look in the top center part here, is that it's actually, there's a control on the gene expression, which happens slowly. But these are proteins called transcription factors. And these transcription factors can sense and actuate control on gene expression. So this is a transcription factor. It can bind onto the DNA and prevent that gene from being expressed. Yeah? But if there's a chemical reaction that happens that produces a chemical that can bind onto that transcription factor, change the shape of it so it let go of the DNA, then gene regulation can go back again. So just like in the thermostat case, we have the transcription factor, which controls, which is an actuator on the gene expression, but also senses the necessary chemical to be able to decide whether to turn it on or off. So that's in essence the control on gene expression. And what I've just described here is control on gene expression is known as transcriptional regulation. This happens very slowly. This can be anywhere in order of magnitude of minutes to hours. But these chemicals can also bind onto the enzymes that catalyze those reactions. It doesn't have to be on the same reaction, it can be on different ones. But this binding happens very quickly. And this can bind onto that enzyme and actually slow down that reaction very quickly. So this is known as allosteric regulation, and this happens on a much faster timescale for shorter term adaptation. So these are the two most abundant forms of control in the cell. The controls are there to address certain problems for the cells to grow and survive. And I want to take you now through some examples of some bacterial control systems that we observe in nature. And I want to highlight the problem that they face at different scales. And that is looking at the molecular level, like at the level of the molecules, at the level of the cell, at population level, and even between the interaction between different organisms, not just different species, but different organisms altogether. And I will show how these control systems are designed and work to beautifully solve that problem. So let's look at the molecular level first, right? Next to the DNA, the most fundamental and important component produced by the cell are called proteins. Yeah? They function to catalyze chemical reactions. They act as sensor regulators, as I just explained. And they are actually the machines which help to copy the code onto the DNA and to translate it, to convert it into a sequence of amino acids which then fold and turn into proteins. But these proteins themselves are composed of a bound string of chemicals produced by metabolism called amino acids. Now, where there's a large change in metabolism, perhaps due to the change in the cell's surrounding environment, a number of different genes may need to be expressed. And the proteins that they encode for, they need to be constructed, right? So there's suddenly there's a lot of demand for these amino acids because you've got to construct these new proteins, right? On the other hand, you may turn off some gene expression. And so you reduce the demand for amino acids because you don't need to produce those proteins anymore. So the question is that how does the cell adapt the production of these amino acids to meet these like ever-changing demand? Yeah. And actually, let's break it down into two types of demand, right? So once the demand is made for the amino acid, right, how do we keep that production consistent? And then secondly, if there's a large shift in the demand, a massive change in gene regulation, some other reactions in metabolism have to be turned on, how do we adapt that production pathway to meet that demand over a longer time scale? So amino acids, you can just think of it in a very simplistic way, but amino acids are produced from what is mainly a single production line, yeah? And let's think about the first problem. So in nature, we observe this solution. Yeah? Here, the end product of that production pathway goes and inhibits, it stops the production in the first part of the production line if there's too much of it. So if there's too much of this end product, it says, well, you don't need to produce me, it's just kind of wasting resources, yeah? Whereas if this drops in concentration, if there's a high demand for it, so this concentration drops, then it doesn't inhibit this anymore and the production line can increase to a certain level. So actually, this is almost exactly analogous to the thermostat example that we saw earlier. But it's turn production on or off to keep that production at a fixed level. That is the way that I see it. Now, with regards to the second problem, that is a little bit more challenging. If there's an increased demand, a massively increased demand, we need to scale up the number of workers along this production line. Let's think about it like that. Think about each of these enzymes that are catalyzing reactions as workers. Yeah? But there's an odd balance to strike here. We need to increase production, but there's a cost. The cost is the constructing of these proteins, these enzymes. We need to recruit more workers, essentially. Yeah. So this is slow and costly. And so you get this sort of trade-off happening here. So you want maximum benefit, but you want to minimize the cost. And there's no sort of solution here, but somewhere this is closest to the optimal, right? That would be great. Yeah. How do we reduce the cost but maximize the benefit? Now there was one of my favorite studies is the study by Chubukov et al. That developed a mathematical model of this production pathway, and they solved this very problem. This is known as a multi-objective. You have two objectives, and you want to solve those simultaneously. And they found an optimal solution, and that optimal solution is that we should increase the number of workers in the reaction that is directly after the part of the production line that is being sensed. So we can sense anywhere along the production line, the state of how much that demand is. If demand is really high, these concentrations will be low. If demand is really low, then these concentrations will be high. So we can use that as a sort of proxy to understand, what is the state? What is the state of the demand? But the general principle that they found that optimally balances these two objectives is to increase the number of workers directly after that chemical that is being sensed. And it doesn't matter actually which chemical. It is always the enzyme, it's always the number of workers after that chemical that's being sensed that we need to increase. So this was found to be the optimal solution. Yeah, it is the low cost investment. It's a nature strategy, is that it's a low cost investment to increase the production. It's such an elegant and robust solution for such a fundamental process in the cell. And of course, there are two levels of control here, a fast-term adaptation and a slow-term adaptation. Now, I'd like to share some of my own recent work on understanding the control between metabolic pathways, not just along a single production line, but looking at multiple production lines, so to say. So bacteria need to make fatty acids, yeah, to construct the cell wall. So they produce these fatty acids, they make the cell wall, and that's how they're able to increase in size physically. Yeah? But if there's fatty acid in the environment, then how can it take advantage of it? Right? Instead of producing it itself, how can it just take it up and use that? So there are two pathways that are associated with fatty acid metabolism. And there's one part of the pathway that decides to take it up and degrade it and use it and turn it into cell wall. And then there's the other one which actually makes it when there's no fatty acid available. So let's look at it like this. When there's none available, this pathway is useless, right? We don't need to invest in proteins than that. And so there's a central controller in all of this, which is called FAD-R. It's just it's another transcription factor, another protein. And that turns off all of these enzymes that are associated with taking up and degrading and using that fatty acid from the environment. But at the same time, it activates the pathway that creates the fatty acids that can be used to grow. But when there is that fatty acid available, it comes in, it binds onto this controller, and essentially it turns it off. So now that can no longer inhibit the expression of these enzymes. So all of these enzymes, all of these workers are turned on, and this pathway is turned off now, right? So it's doing a switching. And so in principle, what you, in the switching, this is a very nice solution, because what it does, it takes all of the resources that you were using to make these, right? It turns these off and turns these on, which means that you're saving resources here, but you're redistributing them, you're reinvesting them here in the uptake. But what if the supply runs out? What if we don't have oleic acid anymore? So we need to switch back to this phase, and we need to switch back quickly. If we don't switch back quickly, the cells can't grow. And instead, they will waste resources just making these enzymes, which are not doing anything. So it's kind of wasting its resources in doing it. Now, we developed a mathematical model to address the question of how to switch. How does the cell switch back quickly? Does it switch back quickly? And the model itself allowed us to discover that the main control feature that addresses this particular problem is what's happening here. This controller actually is regulating itself. Yeah, it's inhibiting its own expression. So if this is at very high levels, it says, okay, you don't need that much controller anymore. But if this is very low levels, it says, okay, make the control. Again, keeping at a consistent level, as we've seen before. Now, when this does happen, what happens is when there is nutrient available, it comes in, it binds onto this controller, and it then sort of accumulates in the cell, because it comes in, it binds. Now this becomes low, so it's not able to repress itself anymore, which means that it makes more of itself. It's now able to make more of itself. But when it does that, then more of that binds onto the chemical that comes in. And so what you get is this kind of cycle of accumulation of this complex of this chemical bound to this protein. And because of this accumulation, this is what creates this quick switchback. Actually, this accumulation, what it's doing is storing that transcription factor, it's storing that controller. And as we store that controller, then when there is none of this nutrient available anymore, then this stored controller can be quickly released to to turn this pathway off and turn that one back on again. And that is what creates the quick switchback. It's such a simple solution, which kills. Oh, I don't want to say kill two birds with the same stone, but in essence, it does that. So you're controlling the amount of expression, the amount of investment that you're doing in this controller. So you're just saying, okay, keep it at low levels, keep it at consistent level. But then again, also have it there, create a storage of it when there is nutrient, so that you can switch back quickly. And we found exactly this. We validated exactly that principle from the experiment. So the more time that we exposed the cell to the nutrient, this is the exposure time, more time we allowed it to be exposed to that, then the recovery time was less, meaning that what this time it took to switch back to this state was less. Yeah, this is the recovery time. This is the exposure time. So you expose it more, recovery time was less. And that is as opposed to if it was regulating itself in a different way, if it was causing its own activation, if it became bound to this, it can't activate itself anymore. And so what happens over time as the cells grow and divide, it just becomes diluted and goes away. So you don't have that storage. And always you have the solution that the time it takes to recover, to switch back, is always much greater. It's a general principle that can be proved mathematically. So in summary, we have a switch to uptake and take advantage of nutrients when it's there. And this so-called negative autoregulation, yeah, allows the cell to store that controller and to quickly switch off the uptake for the efficient reversion back to biosynthesis. So it's investment a bit in the storage to enable the overall lower cost of producing proteins, because it prevents any expression of unneeded protein. Again, such an elegant solution. We now zoom out a little bit more to consider a problem associated with evolution. Yeah. So for long-term survival and adaptation, cells can genetically diversify. And this is costly, because when you genetically diversify, right, those different, they're called genotypes, so those cells which are different in their genetic makeup now, might not grow well in that current environment, right? But it's an investment for the future, because it may grow much better in the changed environment. But the question becomes that when should we increase this genetic diversity? Because if we do it when there's too few cells, when they're not growing very well, it's very expensive, because it takes away from the growing population. You can't grow your population that well. Yeah. If we do it too late, the population is quite homogeneous. It may, it's not diverse enough, and so it doesn't have much benefit. So there's some sort of optimal line in between. You've got to invest something. Yeah. If you don't invest much, there might be a temporary benefit, but when the environment changes, that benefit is not really long term. And again, at the other end, high cost, you can't grow very well, so the benefit is again reduced. So there's an optimal somewhere between. So this is how I'm speculating what the trade-off looks like. Yeah? There's a control system at the molecular level that can solve this problem. Yeah, it's very cool. There was a study from the group of Paulson in Harvard Medical School, and they identified this mechanism where you have this protein produced called ADA. Yeah, what it does, it looks at methylation on the DNA. So when DNA is methylated, it can become damaged, and in repairing that damage, new mutations can be added. Right, so this leads to the genetic diversity. I hope that makes sense. Yeah. However, there is a protein that demethylates the DNA to protect it from DNA damage, and this is ADA. So the ADA is produced; it binds onto the methyl group that's on the DNA, and this complex then ends up coming back and making more of that protein. So it goes and supports, more of the demethylation. Yeah, but ADA again controls its own level. We see this ubiquitous, right, kind of design all over nature, and you see it again and again in all the examples I'm giving. So it's controlling itself. Yeah, and so that's able to allow the repairing and keeping the population homogeneous. Now, yes, so sorry, this can keep the population quite homogeneous. So let's say all of these cells, they're producing ADA, yeah, and they're all sort of similar genotype. Well, almost all of them are similar genotype. Now, it's very interesting that actually the average number of ADA molecules, ADA protein molecules per cell is 1. That's very low. You're thinking, what is that going to do? So if the cells grow and divide very, very quickly, some might not have any left in them, right? So what you end up with is some cells that don't have any ADA. And then when this happens, then the cells can then allow for more, well, this allows for more DNA damage, and the rate of diversification becomes increased. And so this sounds, again, like a very elegant solution. The principle is to diversify when the cells are growing very fast. When they're in a very rich environment, they can grow very fast. That's when they can afford to invest in some of their cells, in some of the population, to be genetically different, to invest for the future, even though those genetically different cells may be at reduced fitness in the current environment. So again, it makes sense, you know, to invest when you have the resources to do that. And again, it's a kind of balance between the cost of producing many less fit cells, but the benefit is that you find fitter cells over a longer time scale, a long evolutionary time scale. So it's a cheap investment, but it's again a very elegant solution, and one that's fundamental for the survival of these bacteria, which have thrived since almost the beginning. Yeah. Now, bacteria also work together at a population level to achieve a common objective. That is to produce chemicals that are useful and important for their survival. And one example of this is the production of biofilms. This creates a sort of microenvironment in which the population can thrive. And many infections do this, like strep throat, cholera in your guts, TB in your lungs, plaque on your teeth. Yeah, unfortunately, we see this a lot before we go to the dentist. So these microbes, these microcosms in which these populations grow, in which they create these, are very important for those populations to thrive, because they can even be, the bacteria that live in those microenvironments, in those biofilms, can even become more tolerant to stresses such as antibiotic treatment, which is why you face problems, especially with TB and even with strep throat to a certain extent, for their treatment. The other examples of synthesizing useful products include producing antibiotics. So there's a whole group of organisms called streptomycins, and actually they produce antibiotics to kill off the competition within the same growth environment. And actually 60% of our antibiotics are coming from streptomyces. So we are harnessing that. Conversely, instead of killing off the competition, what you do is you grow in symbiosis. So you create chemicals that can be of mutual benefit. That, in this case of Vibrio fischeri and Hawaiian bobtail squid, this is a very, very interesting example. So Vibrio fischeri is a bacteria. It lives inside the bobtail squid, actually towards the back. I'm sorry, I'm not sure what this is called. And what happens is that the bobtail squid, it comes out at night. It goes to the top of, it goes towards the surface of the ocean, and there's predators lurk underneath it. When it becomes stressed, it releases a sort of nutrient which allows those bacteria to grow. And as those bacteria grow, because they're using the nutrients from the bobtail squid, they grow, they produce a fluorescent molecule that then fluoresces once you have a large enough population, and sort of camouflages. When you look at the bobtail squid from below, it looks like night. it looks like a starry night. So it camouflages against its backdrop of a starry night to evade it from its predators. So there's a very delicate and interesting, symbiosis going on there. But symbiosis can happen on many different levels, and there are many examples. I just picked that one because it's very, very cool. But the question becomes, so how do these populations do this? How do they communicate with one another to strive for this common goal? How do they decide when to do it? That is even more important. So again, it's an investment to shift metabolism away from growing and to instead make these useful chemicals, because it's very expensive to invest in doing both at the same time. And the solution for this is called quorum sensing. So let me go into this in a little bit more detail. Let's focus on this part of the picture first. There's a lot of stuff going on here, but let's focus on this part of the picture first. So first, the cells are producing a small amount of a chemical called an autoinducer. It's just a chemical that can diffuse out of the cell. It goes into the surrounding environment, yeah, and it's produced at a very small amount. Yeah? So there's a very little investment. Okay? But then when when the cells are able to grow very quickly, that means the population becomes very dense. So there's loads and loads of cells, right? And each one of those cells is producing a small amount, right? So that's a lot of this autoinducer that becomes available in the environment. And because that concentration becomes very high, it effectively can go back into the cell or creates a higher concentration in the cell. This binds onto a controller, which senses it, yeah, and activates gene expression. So it turns on more genes to create more even. So you get this sort of sudden increase of this chemical, this autoinducer, which then creates an even stronger activation. And that activation not only activates, the construction of these autoinducers, but also it turns on expression of other enzymes. And those other enzymes are the ones that go and produce those useful products, no matter what they are. Right? Whether it's a biofilm, whether it's a chemical for killing other competition or creating symbiosis. So that's the essence of how these cells do it. And the question of when, that depends on how much is being produced. So if very little is being produced, right, you need the cells to grow a lot before it then goes and turns on the gene expression and turns on the production of these useful chemicals. And that happens at a certain, population size. Yeah. And for different scenarios, that exactly when that happens is slightly differently, but it's tuned to optimize for that. So again, it's worth investing for the long-term survival. Nature's elegant solution is to invest a little to constantly make this tiny bit of this inducing chemical so as to switch when there are many cells to make that useful product. It's very costly to make a product, yeah? So what this is doing is in fact reducing the overall cost of making the product per cell. Yeah? If there weren't enough cells, it would be really expensive to make this product because each cell would have to make a lot of it. But it's not worth doing that because then it's not able to thrive, it's not able to grow, and the population can then end up dying. If it doesn't invest enough, if it does it too late, then it's not able to take advantage of that micro environment. It's not able to outcompete. It's not able to create the symbiosis. So there's again an optimal somewhere in between. So just to summarize, a lot of these mechanisms can adjust the reactions in metabolism's activity to quickly meet demands. They can optimally balance the production with the cost of expression. They can rapidly recover and to save the cost of expressing unneeded enzymes. And they can temporarily shift from growth to production for that longer-term survival. So nature's strategies, in essence, find an optimal and elegant solution that balances these costs and benefits. When you look at it like that, yeah? So it finds an optimal. It's quite amazing, sometimes how simplistic these solutions are, but also how powerful they are. So we've seen many examples, we've seen some examples of the natural systems now, and how they control metabolism and the objectives of these control systems. The objectives seeming to be that they're designed to be, sorry, let me say that again. And the objectives that those control systems seem to be designed to be elegantly addressing. We've seen those. Now we want to visit a vision of how humankind can harness these amazing capabilities of nature for its own benefit. And in particular, here I want to focus our attention on the capability of bacteria to produce a wide range of useful chemicals. Yeah. So the question becomes, how should we rewire this metabolism, right, to harness the production capability of these cells? And even more importantly, how can we increase the capacity of the chemicals, of the capacity of the cells to produce even more chemicals? If we can address these, then we can develop bacteria for various applications. Yeah? And some of these applications include bioremediation. So what does that mean? That is to help us to sustain a healthy and clean environment that, unfortunately, that we keep polluting as humans. Yeah? And I can give a small example of oil spills that happen in the ocean. They've actually found microorganisms that can break down the long chain hydrocarbons chains in crude oil that can help to break them down. But if there's an oil spill, I mean, those bacteria aren't able to cope with it. So can we increase its capacity? It has the capability, but can we increase its capacity to clean that environment, for instance? We can use bacteria as even live therapeutics. There's a very, very exciting study going on now that I heard about a couple of years ago, but it's really up and coming now. And that is to engineer bacteria to kill cancer tumors from the inside out. So what happens is that you engineer bacteria to produce a cancer-killing chemical, yeah, some chemotherapeutic chemical, and it will grow to very high concentrations. It's actually found that bacteria colonize to very high concentrations in tumor cells as opposed to other tissues in humans. So we can exploit that. And when they grow to very high concentrations, imagine, the quorum sensing scenario, where then you send a signal out that says, okay, burst, yeah, to release these chemicals. The cells burst, they release the chemicals that kill the cancer cells from the inside out. But of course, there are a small number of bacteria left over, and that creates the cycle to do that. And they've shown in mice that it is together with chemotherapy, not just by itself, but together with chemotherapy, it has been shown to reduce the size of the tumor itself. And that was a tumor that was grafted onto the back of mice. Then they showed it for a tumor that was grafted onto onto the liver of the mouse. We can also engineer bacteria with the vision of creating sustainable production of useful chemicals for cosmetics, for pharmaceuticals, even for biofuels. Yeah, and the biofuels is known. We've been doing that for quite some time now. In fact, 30% of some diesels are coming from crops. So, you have the biofuel vision already coming into fruition. Yeah? And this can enable us to create a cyclic economy. And this chemical, products from bacteria, it's a very big economy. At the moment it's 180 billion, but it's predicted to increase by 50% by 2023. So there's a massive demand for it. And so can bacteria be engineered to meet that demand for sustainable, environmentally friendly production of these chemicals instead of polluting production way? So a lot of these visions are currently being looked into, but finding the solutions has been so far by looking for it in nature. they haven't engineered the bacteria to really do those, because the capacity, the capability is there, but the capacity is not. So what are some of the fundamental problems? Yeah. Oh, sorry, before I go into that. So like I said, that people have been looking for examples in nature, yeah, to be able to address these kind of visions. And actually some of them are already there in the products that we use, right? There are bacteria that already degrade the polluting oils, or there are friendly live bacteria in our yogurts. It says live, right? When you pick up a yogurt from the supermarket. So those are bacteria, but they're not engineered, so don't worry about that. But especially for chemical production, right? The capacity is still a massive issue. It's not economically viable. But when we engineer the cell and attempt to rewire its metabolism, there's a number of challenges and open problems. And a lot of these I'm very interested in looking into for my research as well. And I wanted to share these with you. So one of those fundamental problems is creating a metabolic imbalance. If we rewire metabolism towards, chemical production, we're kind of stealing resources. We may be stealing resources away from growth, but not only that, but also creating the energy that we need to power up these machines in the cell. We may be stealing away resources from making the proteins, which are fundamental to all the processes, right? You've got to share some resources. It's a competition for limited machinery. Yes, there's a competition for production and growth, but there may be an optimal for the amount of chemical that you can produce per time. Yeah, and that's known as productivity. So there may be this trade-off between how much product you can make in growth, but there's an optimal point somewhere in between for maximizing the amount you produce per time. But then even if we redistribute metabolism to here, how do we do it? Maybe we need to put a control somewhere. We should learn from nature solutions. We need to repurpose them. But where do we put these controls in metabolism? How should they function? What should the parts and interactions be? What should that circuit look like? Yeah. And then another fundamental problem is controlling genetic diversity. I've already explained how fundamental this is to the survival of cells in a natural setting. But for human purposes, this is no good, right? Because it will mean that there's a subpopulation of bacteria that are not really producing the product that we want, and so it's a suboptimal production. Yeah, and we've got to sustain this production robustly. We can't get the cells to move away from how we've engineered it to function. Now, I've already started hinting on the fundamental root problem that's underlying all of these problems. And that is that the human-centric objectives often oppose those that are in nature. And when we try to fight against nature, it's very difficult. So it's my humble opinion that we must instead learn from the principles of control systems and their designs in nature and repurpose them to address these problems. And I'm very strongly believing that a multidisciplinary approach, together with mathematical models and chemists and experimental biologists, this approach together is the way to be able to solve these problems. So as an example, I just wanted to propose some potential solutions to these problems that I've highlighted up here. Yeah. A promising strategy to overcome this trade-off between whether you should produce or whether you should grow, and which may even address metabolic imbalance, but that's still an open problem, yeah, is to first grow, then produce. So we should have a switch that first allows it to grow and then switch to production. That's what happens in nature, right? As we saw in the examples of the quorum sensing. Why is that a good solution? Well, already there's been theoretical evidence. There's already evidence that such a strategy can result in much higher product made within the same time frame. So imagine this is the time, right? And this is the amount of product. If each cell, produces that product consistently, it's going to grow less because it's taking away from the growth, right? And so by this time, you'll have like 4 cells and 4 products. Yeah. But here, if you first let them grow, they'll grow very quickly to a high population and then switch to growth. There might be a small leaking of the amount of production. That's fine. That happens. But in the end, what you have is you have far more molecules. You have 8 cells. You have, each of them producing twice as much. So you have 16 molecules of the product as opposed to 4 within the same time frame. So productivity can be much higher, yeah? Especially when that trade-off between growth and production is very severe. That's a potential solution, and that has already shown promise. So there's been studies from 2015 and have implemented them experimentally as well, from 2018 and beyond, and proven this. What about the design of the switch? How should we do that? So actually, this falls very much into the remit of my own work. And what I'm showing here is coming up in my upcoming paper. But in brief, we can develop mathematical models of these control systems and start to play around with that model and try to understand how we should design that circuit, what that circuit should look like, what its function is, and how to achieve the function that we want. So in this case, what I've developed here is looking at a control system which allows us to induce it with a cheap inducer, with a cheap chemical, and then switch it to away from growth and into producing the chemical of interest. But if we take away this inducer, usually it'll switch back very quickly. That's what my previous study showed. But in this case, it won't switch back. So what happens is that you add in an inducer, you go from the low state to the high state, that is the production state. Yeah? And now, if you reduce the inducer, you can't switch back. You can't fall back onto the low state. So this is known as an irreversible switch. Yeah, if you're interested, look out for that upcoming in the next couple of months. And we can use these mathematical models to optimize, our objectives. So we've tried to discover the optimization problem in nature, but we can optimize as well using these mathematical models of the objectives of our objectives of interest. So that may be a way forward to help us to design these switches, as opposed to just trying them out in the lab, which is a very long and laborious process. What about harnessing quorum sensing? I mean, the solution is there in nature already, yeah? And studies have started to do this in the past 4 years now, yeah? There have been 2 or 3 groups of metabolic engineers that have looked into this particular solution, and they have harnessed that. There are some problems associated with this, but it's amazing that we can exploit this quorum sensing system to be able to do that switching. What about the control on genetic diversity? This is one of the most fundamental problems and hardest problems in engineering biology. And there are loads of people in different areas. So when genes are, one of them is in the stochastics as well. That is, gene, when they're expressed, they're not expressed constantly, consistently in a very smooth way. It's a kind of burst of expression. Yeah. And when this burst of expression happens, then there can be fluctuations. And those fluctuations can lead to the cell's behavior jumping around a bit. And so they say, okay, what if we help to hinder that fluctuation, right? Can we, delay the onset of genetic diversification? The answer is yes, but how exactly that happens is very, very difficult. Yeah? There's a recent paper from, I say recent, it's 2016 now, from one of my collaborators, and it's a very nice solution. So they harness, there's a mechanism called that allows for antibiotic resistance, and they harness that to select for strains that are higher producers. So when we're growing these cells in a container, yeah, We want them to produce this product, but not all of them are producing it because the environment is very heterogeneous, it's very mixed, right? And so the cell's behavior will fluctuate as well. That will become very mixed. So actually what we end up with is about 30%, 15% of the population producing that product at a very high level, and others not really producing much at all. That's no good, right? So how do we select for those higher producing cells so that we bias our population becomes more homogeneous on the higher producing side? And their solution was to sense that product through a transcription factor, through a sensor, and cause an activation of a protein that just spits back out antibiotic once it comes into the cell. That makes the cell more antibiotic tolerant, and so it lives. If there wasn't enough of this product, it won't produce this, it won't spit the antibiotic back out, and so it will die from it. Yeah? And so what happens is you're selecting for the higher producing strains in that way. So this is a potential solution, although the actual mechanism itself, it's hard to scale it up because adding antibiotics to a huge container on an industrial level is very expensive. But similar kind of solutions can be taken forward. This was one that was harnessed from nature itself. So just to summarize everything in the talk, throughout the whole talk, I'd like to point out that we saw a few examples of beautifully designed and optimized control in nature. But being human-centric and selfish in our needs, nature's capabilities don't match the capacity for human need, because our objectives are not, equal to those of nature, of course. Yeah, where they are, we can exploit those, but most of the time it's not. And so it is my humble opinion that we should learn from nature and distill these design principles that we observe in nature, and then repurpose them for human needs, just in the example that I showed before. But that requires it requires an interdisciplinary approach. It requires, the amalgamation of different disciplines. And one of the verses of the Holy Quran I'd like to point to that inspired what I think is inspiring the same vision. And again, this is up for discussion because this is my own interpretation. Yeah, is in verse 30, is in chapter 30, 31. Lamia says that, so set thy face to the surface of religion as one devoted to God, and follow the nature made by Allah, the nature in which He has created mankind. There is no altering the creation of Allah. That is the right religion, but most men know not. So there's a lot of richness in this amazing verse, right? I don't really know how to say it, but it says there's no altering the creation of Allah, the nature in which He has created mankind. For me, when I read this, it's like as if I'm understanding that as learn from nature, right? Don't go against it. That's what I understand by no altering the creation of Allah. Yeah? Learn from nature, that's how we can harness it. And again, it's all in the matter of seeking that knowledge, but most men know not, so seek the knowledge for it. And Khalifah Rabi Rahmatullah, in his book, Revelation, Rationality, Knowledge and Truth, where he talks about genetic engineering, I just wanted to highlight something that stood out to me, where he's talking about the ethics of genetic engineering. And he highlights that some scientists argue that we should develop genetic engineering as a field in its fullest extent so that we may discover the secrets of creation. And they believe that, and those scientists believe that technological developments in this field will brighten the future of man. And I think I fall somewhere in this area as well, though, I mean, this is much more stronger a statement, fullest extent. But I myself would sort of fall into that area as well. There are other verses of the Holy Quran that have struck my attention. There are many, many more, and I'm just highlighting a couple. Yeah, but they fall kind of into three categories for me. One is the looking at the perfection in creation. Yeah, what Alamia is sort of pointing is giving evidence that points to him, right? But in that evidence, that is the observation that we can make scientifically. These are verses that Professor Abdus Salam gave during his Nobel Prize speech. But this one is interesting, where Al-Amir says, again from chapter 30, verse 28, he it is who originates the creation, then repeats it, and it is most easy for him. He is the most exalted state in the heavens, in the earth, and he's the mighty, the wise. Repeats it. What it means is that that solution is repeated again, right? The mechanism may be slightly different, but the objective, the solution, the optimization problem, everything is repeated over and over. So again, remember this negative autoregulation that I keep talking about? That was a repeated solution over and over. And it works on different scales. I showed it on all of the 4 scales that I was talking about. So that's how I've understood this as well. Of course, there are verses around urging to seek knowledge. That is, we should pursue, well, in my opinion, I would like to say we should pursue scientific discovery. But there's also a warning about, that becoming vain in our desires, that Shaitan will, that Satan will sort of incite us to maybe become carried away. That we will alter the creation of Allah. Maybe there are dangers there. We could create, biological warfare, for example, right? That's very dangerous. So I'd just like to end by acknowledging, all the people that I worked with through the years, those that have supported me, enabled me to gain knowledge in various subjects and areas, across different disciplines as well. And I've learned so much from experimentalists. I cannot even begin to thank, the abundance of knowledge that I've learned from them, and what has driven my modeling work, theoretical work as well. So I'd like to open up for questions and discussion, and there are a number of questions, so please read through these. I won't read through them. Read through these. These are questions that I pose to you, and I'd like to get your feelings and opinions and comments on those questions as well. Jazakallah.